Dunlop from the Ministry of Natural Resources, and she's going to talk to us about next generation Lampricides.
Okay, good morning. So my name is Dunlop. I'm with the Ontario Ministry of Natural Resources. And my co-author on the presentation today is Nadia Carmencini from the USGS Upper Midwest Environmental Science Center. And I'll say that the second part of this presentation that really focuses on the next generation Lampricides is really Nadia's project, but she couldn't be here to present that work. So I hope to answer 3 questions in the presentation today. One is just what is resistance? Why should we care about resistance? And how can the development of next generation Lampricides help? So to start off with, I wanted to provide a definition of resistance so we're all on the same page. And very simply what it is a genetic-based change in susceptibility of a pest population to control measures. So in the case of sea lamprey, of course, we're talking about a genetic change in the sea lamprey that allows them to survive TFM treatment. You've probably heard about resistance developing in other systems, for example, in agricultural systems, when insect pests develop resistance to pesticides. You've probably all heard about, concepts like antibiotic resistance. So it all kind of basically means the same thing. And it has the same results in the end, and that is reduced effectiveness of the control measure and ultimately increased pest population abundance. So I just wanted to walk through and give you a sense of how resistance can develop. So we can think about this as our sea lamprey population. The green individuals there are all our kind of like normal sea lamprey that when exposed to the pesticide, in this case TFM, they die as a result of that exposure. However, some sea lamprey we know do survive treatment. And in the sea lamprey control circles, I hear a lot that upwards of 90% or even like 90% of stream treatments are effective. So 95% of the sea lamprey in the stream are killed. So there are some residual lamprey. And so if we're talking about resistance, what we're meaning here is that these lamprey have a genetic propensity to survive that treatment. And so we call these individuals resistant individuals, so they have resistance alleles. And resistance can arise in a population through different means. One is that you can just have some standing genetic variation in your population. So this is variation that would have come in with the founding population into the Great Lakes. And so there may just be, a few resistant individuals out there that, are very rare, so we haven't really detected them, but they are out there. As well, resistance can arrive through novel mutations. So this is just by random chance, a change in the genome of an individual can allow them to survive that treatment. And the other way that resistance can come into a population is through migration. So if we still have some migration from the native range, this could introduce that genetic variation and maybe just by chance bring in a resistant individual. So because these resistant sea lamprey, in this case, are able to survive treatment, they have a higher fitness. So they're more likely to survive, to make it out into the lake, and eventually to reproduce. And so what happens is over time, we see that the numbers of these resistant individuals increase because of that increased fitness. And so now we have a population that is mostly made up of sea lamprey that can survive that treatment, and only a very small proportion that sort of continues to behave as we would like. And you even see in some agricultural systems, for example, resistance can completely spread through a population to absolute fixation. And this can lead to complete crop failure. So it's a really serious issue, and it's one that needs to be taken seriously. Of course, we're all interested in just how long does it take for resistance to develop. So there's a number of factors that can influence that. One is generation time. So obviously like an insect or a microbe, it's going to have a really fast generation time. And so you can see resistance develop within a matter of days or weeks or months. The C. lamprey, of course, has a longer generation time, so it's going to take decades for resistance to develop. Fecundity also matters, so a very highly fecund species can have resistance develop sooner, so this is not good news because C. lamprey do have a very high fecundity. Gene flow matters, so if there's gene flow coming in from other populations, or how much mixing there is among populations can influence the speed of resistance development. The genetic nature of resistance, so whether we're talking about a recessive trait or a dominant one, or multiple gene loci being involved in conferring resistance. The selective pressure from the pesticide, so this has to do with more or less like the mortality rate of the pesticide. And then finally, fitness costs. So sometimes what you see is when an individual is resistant, so they have this genetic propensity to survive treatment, this comes at some other cost. So something else in the animal's life history is influenced. And so whether those costs exist can influence the speed of resistance development. Okay, so why should we care? I mean, I think it's pretty obvious, but essentially, if resistance were to develop to TFM, TFM would just not work as well and may even not work at all. And so we just saw a really good example from Ben's presentation of kind of what might happen if TFM were no longer effective, right? It would almost be like we couldn't use it anymore. And so this would ultimately result in increased numbers of parasites and, of course, would threaten our valuable fisheries resources. So I would say, resistance does pose a serious risk to the sea lamprey control program, just as, other risks exist, like climate change or the loss of the social license. This is also posing a risk, and it's something that we do need to think about and address. Okay, so what is the science telling us so far about resistance? First and foremost, we know that enough generations have passed and the selective pressure from TFM is sufficiently high for resistance to develop in theory. So, we know the number of years that Lampricides have been applied, we know the generation time of a lamprey, and so when we put those numbers together and we look at examples from other systems where resistance has developed or where rapid evolution has occurred in fish populations, we know that theoretically enough time has passed. So this just means that this is a problem now and it's something we need to think about now. However, the really good news is that full-blown resistance, physiological resistance, has not yet developed. This is based on toxicity testing that's been done at Hammond Bay over time, where we haven't seen a change in the LC 90 or LC 99, so the lethal concentration required to kill our sea lamprey. That hasn't changed over time. We would expect that it would have changed if full-blown physiological resistance had developed. And also we just know from sea lamprey control folks out in the field, they're not seeing huge numbers of larval lamprey surviving treatment. So this is really good news. This means it hasn't happened yet and we still have time to address this. We do, however, see some red flags, and so this is where we really need to be taking a closer look. So some genetic analysis has shown some potential early warning signs of resistance. And so these are experiments done where they took sea lamprey from the native range and then also sea lamprey from the Great Lakes, and they exposed them to TFM. And what they found were, although there was no difference in the lethal concentrations required to kill these lamprey, so again, no physiological resistance, there were differences in gene expression between these two populations. And the worrying thing was that these differences occurred at gene loci that are important in oxidative phosphorylation, which is related to the mode of action of TFM, which works to uncouple oxidative phosphorylation. So some red flags here that we have to look out for. Okay, so what are we doing to address the risk of resistance? Well, there was a resistance workshop held back in 2014, and there was two publications that came out of that, mentioned down below if you're interested in checking those out. But a big development out of that workshop was the initiation or the development of the next generation Lampricide project, and that's what I'm going to be speaking about in a moment. We also had a second resistant workshop back in 2023, and we're still working through some of the outcomes of that workshop, but essentially what we're doing there is developing options for monitoring for resistance, so we can keep our eye on this. And of course, there's been ongoing research funded by the Commission on this topic. Okay, so the Next Generation Lampricide Project, what is it? Well, basically the purpose of this is to seek out and determine if there's another lampricide out there that could either replace TFM or supplement the use of TFM. So back in the 1950s and 60s, Applegate's experiments, over 5,000 chemicals were actually tested at that time, and that's what led to the discovery of TFM and nitrosamide. However, there's many other chemicals in there that could potentially be toxic to sea lamprey. As well, since those earlier experiments, millions of more chemicals obviously have been developed, and so there's sort of a whole potential there within sort of the knowledge that we have about these other chemicals for there to be another lampricide. So basically, the goal behind this project is to find a new lampricide that could potentially have a novel mode of action, so something that's different than the mode of action of TFM, is specific to sea lamprey, is cost-effective, and is also safe, green, and socially acceptable. So how could the development of a new lampricide help with resistance? Well, what we know from other systems where resistance has developed is that reliance on a single pesticide just increases the chances that it's going to happen. Furthermore, we do supplement, as Bruce was saying, with nitrosamide, but TFM and nitrosamide actually have a similar mode of action, so they're both acting to uncouple oxidative phosphorylation. So that just means you're sort of, in a way, kind of using the same pesticide, although there are differences. So finding a new lampricide with a different mode of action would reduce the chances of resistance if we were to use that alternative pesticide, but also would obviously provide us a backup option if resistance to TFM were to develop. And of course, it can take years to develop a new pesticide, so this is why we need to be working on this now, before resistance develops. Okay, so there's 3 different stages to this project. The first is doing what's called in silico and in vitro chemical screening. So in silico screening just refers to computer. Like, I didn't quite understand what that term meant, but Nadia explained it to me. It's like, oh, okay, I get it. But basically what it means is like screening existing chemical databases. And then in vitro chemical screening means actually developing cell lines for different fish species, and testing toxicity of the chemicals on those cell lines. The second stage involves doing in vivo testing, so this is now where you're testing on whole organisms. And then the third stage is the really comprehensive and very specific set of regulatory studies that you have to do in order to eventually bring a new lampricide and use it in the field. Okay, so I'm just going to provide a little bit of an update on the progress in these different stages. So for the in silico or computer screening that's being done, basically what you do is you search existing databases, and ideally what you need to find is paired chemical information, so information about the chemical structure, and also toxicity data. And you need to have both of these in order to say whether that chemical could potentially be effective against sea lamprey. Now, unfortunately, when they search through these existing databases, there really isn't a lot of toxicity data out there for sea lamprey, and so they had to look to these earlier experiments by Applegate that was done in the 50s and 60s, and they ended up going to Hammond Bay and actually entering in a whole bunch of these records and using that to search for potential alternative lampricides. And so through that search, they found 16 chemicals from 14 different classes that were selectively toxic to sea lamprey. So now they're working to better understand the mode of action and to do further testing of the cell lines. So for continuing on in that stage, some of their progress in this in vitro testing, so testing the toxicity of these 16 new chemicals that they found, on cell lines. So what they've been doing is they've been developing cell lines, or trying to, for 5 different fish species that have different tolerances to Lampricides. Unfortunately, after 2.5 years of testing, they could not develop cell lines for sea lamprey. And so instead, they're using lake sturgeon and rainbow trout cell lines to do this testing further. So what this involves is exposing those species, their cell lines, to TFM, and then testing the function of the cells in a few different areas. And to start with, they were testing toxicity to TFM. So seeing, like, could you replicate what we know of TFM before then moving on to these other chemicals. In a nutshell, what they found here was that when you test the gill cells for rainbow trout and lake sturgeon, they actually show very similar toxicities to TFM. Now, this is not what they were hoping to see, because we know that from prior work done on in vivo testing, so whole organisms, from Bogart et al.'s work in 2003, that we would have expected to see differences between sturgeon and rainbow trout in their response to TFM. We know that sturgeon are more sensitive. So unfortunately, what this means is that the gill cell lines sort of aren't really working well in this case. And so next, they're going to be looking at liver cells to do this work. Okay, so that's it. So basically, important progress has been made on this project, but more work is clearly needed in order to develop a next generation Lampricide and reduce the risk of resistance. And I just wanted to close by saying that, unfortunately, Nadia's work, it does, with the recent staff reductions and some of the questions about funding, these issues are sort of putting this next generation project at risk. So just a message she wanted me to convey. Okay, thank you.
Thanks, Erin.